Hi, I am Anuj Modi, Assistant Professor at United Institute of Pharmaceutical Sciences, Sagar, MP, with an interesting and learning episode of B.Pharm on the important topic phytochemical screening 4, that is radio tracer technique and their utilization in elucidation of biosynthetic pathway in plants, which is one of the important unit of section pharmacognosy B.Pharm 6th semester. This is prepared by me and Dr. Bimal Kumar, Professor and Head, Department of Phytopharmaceuticals and Natural Product, Institute of Pharmacy, Nirma University, Ahmedabad, Gujarat, India. Let's start our episode while taking a look on that what we are going to learn today. Module 1, Introduction, that is elucidation of biosynthetic pathway and tracer technique. Module 2, radioactive tracers and requirement for tracer technique. Module 3, method for elucidation of biosynthetic pathway. Module 4, application of tracer technique in biogenetic studies. Module 1, introduction, elucidation of biosynthetic pathway and tracer technique. The ever-expanding knowledge of natural products leads to curiosity concerning the transformation and function of biological molecules. Living plants are considered as biosynthetic laboratory of primary metabolites which are used by the plants for its own growth and development, leading to the formation of secondary metabolites. These secondary metabolites are waste product of the plants but produces specific pharmacological effects in human beings. Different biosynthetic pathway involved in the metabolism of primary metabolites or biosynthesis of secondary metabolites are sigmic acid pathway used for biosynthesis of aromatic amino acids, mevalonic acid pathway used for biosynthesis of terpenoids and steroidal compounds, acetate pathway used for biosynthesis of fatty acids. Before knowing about the tracer technique, we must have to understand the difference between biosynthesis and biogenesis. Biosynthesis means formation of chemical compounds by living organism, while biogenesis means production or generation of living organism from other living organism. Secondary metabolism now provides an essential bridge between the physical and life science. The advantage of understanding the biosynthetic pathway depends on various specialized techniques from wide range of disciplines. The recent techniques used for elucidation of biosynthetic pathway are use of isolated organ, grafting methods, use of mutant strains, and tracer techniques. Tracer technology, now widely employed in all branches of science, had its origin in the early part of the last century, when it was realized that elements exist with the identical chemical properties but with different atomic weights. Such isotopes may be stable or unstable and decay with the emission of radiation. If it is possible to detect these isotopes by the suitable means, then they can be incorporated in presumed precursor of the plant constituent and used as a marker in biogenetic experiment. Tracer technique can be defined as a technique which is utilize a labeled compound to find out or trace the different intermediates and various steps of biosynthetic pathway in plants at a given rate and time. Or in this technique, different isotopes, mainly the radioactive isotopes, are incorporated in presumed precursor of the plant metabolites and are used as a marker in biogenetic experiment. Now What is the significance of tracer technique? There are many significance of tracer technique like high sensitivity, applicable to living system, wide range of the isotopes are available, more reliable, easy administration and isolation procedure, gives accurate results if proper metabolic time and technique applied. Different tracers can be used in different study. Module 2, radioactive tracers and requirement for the tracer techniques. A number of isotopes of common stable elements are found in the nature. These isotopes can be artificially produced through the nuclear reactor and accelerator facilities. The radio-labeled compound can be prepared by the use of two types of isotopes. First, radioactive isotopes. Second, stable isotopes. Radioactive isotopes, for example, tritium, carbon 14, sodium 24, potassium 42, sulfur 35, phosphorus 35, iodine 131. They decay with the emission of radiation. Carbon and hydrogen labeling are used for the biological investigation. Sulfur, phosphorus, alkali and alkaline earth metals labeling are used for metabolic studies, while nitrogen labeling are used for studies on protein, alkaloids and amino acids. Second one, stable isotopes used for the labeling of possible intermediates in biosynthetic pathway in a similar way as radioactive isotopes are used. For example, hydrogen 2, carbon 13, nitrogen 15, oxygen 18. Usual methods for the detection are stable isotopes, mass spectroscopy used for the detection of nitrogen 15 and oxygen 18 labeling. NMR spectroscopy, it is used for the detection of hydrogen 2 and carbon 13 labeling. Now we will see what is the criteria for the selection of radioactive tracers used for the labeling of the precursors. The starting concentration must be sufficient to withstand with the dilution in the course of metabolism. Second one, proper labeling. For the proper labeling, physical and chemical nature of the compound must be known. Third one, labeling compound should not involve in the biosynthetic reaction. Fourth, labeling should not damage the system to which it is used. Some important properties like half-life and emission of isotopes are given in the table. Tritium, it have the half-life 12.43 years and emits beta particles, while carbon 14 have the half-life of 5730 years but it emits weak beta emission. Sulfur 35, it have the half-life of 87.4 days and emits beta emission. Phosphorus 32, it have half-life of 14.3 days, emits beta emission, while iodine 131 have the half-life of 8.04 days and emits gamma emission. It has some limitations like kinetic effect, chemical effect, radiation effect, radiochemical purity and high concentration of distorting the results. Requirement of tracer techniques. Tracer techniques mainly involve three steps. First, preparation of labeled compound. Second, introduction of labeled compound into a biological system. Third, separation and determination of labeled compound in various biochemical fractions at a specific time interval. We will study each step one by one. First step, that is preparation of labeled compound. Method used for the labeling of the compounds depending upon the type of radioisotopes used. For example, carbon 14 labeling. The carbon 14 labeled compound produced by growing chlorella in atmosphere of carbon 14 labeled carbon dioxides. All carbon compounds become 14C labeled. 14C is commercially available as barium carbonates. 14C emits beta particles. Tritium labeling. Tritium labeled compounds are commercially available. Tritium labeling is affected by catalytic exchange in aqueous media by hydrogenation of unsaturated compound with tritium gas. Tritium is a pure beta emitter of low intensity and its radiation energy is lower than carbon 40. Other isotopes labeling. Radioactive isotopes of sulfur, phosphorus, iodine are commercially available, as relatively few naturally occurring molecules use these elements exist in the nature. Second step, that is introduction of labeled compound. When radioisotopes labeled precursor are fed to the plants, precautions should be taken to that, like the precursors should react at necessary site of the synthesis in plant. Second, during experiment, plant should synthesize the compounds under investigation. Third, the dose given in the short period. Fourth, different methods are used for the incorporation of radio-labeled precursor in biological system. Like first, root feeding. The plants in which roots are the biosynthetic sites, for example, tobacco. Stem feeding. Substrate can be administered through the cut end of the stem immersed in the solution. This method is not suitable for the latex containing plants. Direct injection. This is suitable for the plants with the hollow stems like umbelliferous fruits and opium. Infiltration or wick feeding. Suitable for the plants rooted in soil or other supports without disturbing the roots. Floating method. When a small amount of the material is available, this method can be used. This technique is used in conjugation with vacuum infiltration to remove the gases. Spray technique. Compound has been absorbed after being sprayed on the leaf in aqueous solution, for example, steroids. Third point, that is the third step of separation and detection of compounds. Separation of compounds depends upon type of material or tissue used. For example, soft and fresh tissue can be extracted or separated by the use of maceration and infusion, while hard tissue, decoction method or hot percolation methods are preferred, and for the unorganized drug, maceration with adjustment can be done. The nature of the solvent used for the extraction depends upon the nature of secondary metabolite present. For the extraction of fat and oils, non-polar solvents are used, while for the extraction of alkaloids, glycosides and flavonoids, slightly polar solvents are used. For the extraction of phenols, highly polar solvents are used. Radioactivity of labeled compound can be determined by using different types of the detectors. First, gas ionization chamber. They use the ionization effect of the radiation upon a gas-filled sensor. If particles have enough energy to ionize a gas atom or molecule, the resulting electrons and ion causes a current flow which can be measured. Geiger-Müller counter. A Geiger-Müller counter is a device which is used for the detection and measurement of all types of the radiation, that is alpha, beta and gamma. Basically, it consists of pair of electrodes surrounded by gas, that is usually helium or argon. The electrodes have high voltage across them. When radiation enters the tube, it causes ionization of gases. The ion and electrons are attracted to the electrode and an electric current is produced. The scaler counts the current pulses and one obtained a count whenever radiation ionizes the gas. Few advantages like relatively inexpensive, durable and detected all types of the radiation. Along with these advantages, this have some disadvantages also, like they cannot differentiate which type of the radiation is being detected. Second, they cannot be used to determine the exact energy of detected radiation and have a very low efficiency. A scintillation counter is used for detecting and measuring ionization radiation, mainly beta emission, by using the excitation effect of incident radiation on a scintillator material and detecting the resulting light pulses. It consists of a scintillator or which generates photon in response to incident radiation. Second, a sensitive photomultiplier tube detector which converts light into an electric signal and electronics to process the signal. Scintillator consists of a transparent crystal, usually a phosphorus or organic liquid, which is called as liquid scintillation counting, that fluoresces when struck by ionization radiation. Different crystals are used for the detection of different types of emission radiation. For example, calcium iodide is used for the detection of protons and alpha particles. Sodium iodide, it contains a small amount of the thallium, is used for the detection of gamma waves. Zinc sulfide is widely used as a detector of alpha particle. Lithium iodide is used as a neutron detector. This detector has certain advantages like widespread detection. Second, can measure both the intensity and the energy of incident radiation. Third, more sensitive than Geiger-Müller counter. Next detector is autoradiography. Autoradiography is nothing but an image on nuclear emulsion, that is silver halide, or on the X-ray film that is produced by the pattern of decay emission, that is beta or gamma emission from a distribution of radioactive substance. The specimen is kept in a contact for a period of time, causes darkening of X-ray sensitive film or silver bromide solution, which is also known as autoradiogram. Autoradiograph gives distribution pattern of the labeled radioisotopes in the specimen. You can see in the figure the detailed steps involved. Autoradiographic technique can be applied to a morphological plant part or to a microscopic section of the specimen. A wide variety of procedures are used in autoradiography, like contact radiography. Second, gel method. In case of the contact radiography, sample and photographic films are held in contact during the period of exposure, while in case of liquid emulsion method, which is also known as gel method, gel is poured over the specimen separately to distinguish between beta particles emitted from carbon-14 and tritium. Next detector is radio paper chromatography. Radio paper chromatography mainly use the tracer studies of metabolic pathway of amino acid and carbohydrate. This technique is also applied for the location of radio-labeled phosphorus product in the study of ATP and ADP. Next detector is Mass spectroscopy. Mass spectroscopy is actually an analytical technique that measures the mass to charge ratio of charged particles. It is used for determining the masses of particles, for determining the elemental composition of a sample or molecule, for elucidating the chemical structure of molecule such as peptides and other chemical compounds. Next detector is NMR spectroscopy. NMR spectroscopy is a research technique that exploits the magnetic properties of certain atom nuclei to determine physical and chemical properties of atom or molecule in which they are contained. It is based on the phenomena of nuclear magnetic resonance and can provide detailed information about structure, dynamics, reaction states, and chemical environment of the molecule. Dear friends, till now you have gained a complete idea about elucidation of biosynthetic pathway, radioactive isotopes, and requirement of research technique. If you have missed any of the module or content, you may log on to our website, that is www.cec.nic.in, to download videos, FAQs, LORs, and other content so that you can remain updated this chapter. Now let's move ahead to our next module, that is about different methods used in the tracer technique. Module 3, that is method used in the tracer technique. There are 4 different methods generally used for the elucidation of biosynthetic pathway are: first, precursor product sequence; second, competitive feeding; third, sequential analysis; fourth, use of stable isotope. Now we move towards each method one by one. First, that is precursor product sequence analysis. In this technique, the presumed precursor of the constituent under investigation in a labeled form is fed in the plant for a shorter period of time, and after suitable time period, constituent produced in the plant is isolated, purified, and radioactivity has been determined. The radioactivity of isolated compound alone is not usually sufficient evidence that the particular compound feed is a direct precursor, because substance may enter the general metabolic pathway and form they may be randomly distributed through a whole range of product. In such case, further proof can be obtained by double and triple labeling experiment. For example, biogenesis of anabasin alkaloids from doubled label lysine proved that which nitrogen of the lysine molecule was involved in the formation of piperidine ring of anabasin in Nicotiana glauca. Non-equivalency of two nitrogen is demonstrated by feeding experiment with radio-labeled nitrogen at carbon number 2 and carbon number 6 of lysine molecule separately. Study results conclude that nitrogen attached to the carbon 2 was involved in the formation of piperidine ring of anabasin rather than nitrogen attached at the carbon number 6. This can be clearly seen in the figure. Second method, that is competitive feeding. In the biosynthesis of secondary metabolites, a number of precursors seems to be involved in the normal route of synthesis. Competitive feeding experiments have the great value to determine which of the available intermediate are used as a precursor by the plant. Consider a biosynthesis of compound C from compound A may be either via intermediate precursor B or B'. In such condition, both the intermediate precursor, that is B and B' are fed separately along with labeled A. If the radioactivity is observed in the group receiving B and not in B' receiving group, path A to B B to C may be concluded as a correct path for the synthesis of compound C. Example: tyrosine is a precursor for 3,4-dihydroxyphenylpyruvic acid and 3,4-dihydroxyphenylethylamine for the synthesis of norlutenicillin, which produce morphine through reticulin. 3,4-dihydroxyphenylethylamine produced from tyrosine via 3,4-dihydroxyphenylalanine, that is also known as DOPA. It was considered as another precursor or intermediate in the synthesis of 3,4-dihydroxyphenylpyruvic acid. But by labeling experiment and competitive feeding, it has been confirmed that tyrosine gives directly 3,4-dihydroxyphenylpyruvic acid without any intermediate. This can be seen in the figure. Next method is sequential analysis. This method is mainly used to determine the sequence in which various correlated compounds become labeled, which can be done by growing the plant in the atmosphere of carbon-14 labeled CO2 and then analyze the plant at a given time interval. This sequential method have wide range of application, that is carbon-14 labeled CO2, and sequential analysis has been successfully used in the elucidation of carbon in photosynthesis. Second, determination of sequential formation of opium, hemlock and tobacco alkaloids. The mentha piperita, when exposed to carbon-14 labeled CO2 for about 5 minutes, provides the evidence of the probable biosynthetic sequence of menthol, that is piperitone to menthone, then menthone to menthol. Sequential analysis method also employed for the investigation of biosynthesis of morphine from thiabine in Papaver somniferum. Sequence of events takes place as follows, that is thiabine is converted into the codeine and codeine gets converted into the morphine. Last method, that is use of stable isotopes. Although the isotopes like H2, carbon-13, nitrogen-15 and oxygen-18 have lower natural abundance but can be used in a way similar to the radioactive isotopes. The usual method of detection are mass spectroscopy, which is used for nitrogen-15 and oxygen-18, while nuclear magnetic resonance spectroscopy is used for the detection of hydrogen and carbon-13. It is now possible in certain instances to determine the structure of natural products and establish a biogenetic pathway by incorporating C13 precursor and using C13 NMR for detection. The use of stable isotopes of C13 has greatly eased the study of role of acetate in polyketide pathway. Module 4, that is application of the tracer technique. Tracer technique has wide range of the application in biogenetic studies. For example, that is first, carbon-14 and tritium labeling mevalonic acid is used to study the cyclization of squalene. Second, interrelationship among 4-methylesterol and 4,4-dimethylsterol has been determined by using carbon-14 labeled acetate. Third, elucidation of terpenoid biosynthesis by chloroplast isolated in organic solvent by the use of carbon-14 labeled mevalonate. 4. Study the formation of cinnamic acid in pathway of coumarin from labeled coumarin. 5th, origin of carbon and nitrogen atom of purine system by use of carbon-14 or nitrogen-15 labeled precursor. 6. Study the formation of scopolamine by the use of labeled phenylalanine. 7. Use of calcium-45 as a tracer shows the uptake of calcium by the plant from the soil. 8th, ammonium phosphate labeled with phosphorus 32 is used to study the uptake of phosphorus by the plants. Study results proves that phosphorus reaches first in lower part of the plant, then the upper part of the plant, that is branches and leaves. We can conclude here that tracer technique is an important tool to determine the various steps or intermediates involved in the biosynthesis of secondary metabolites. This technique is also used to study the location of particular compounds and metabolism. With all these information, here we comes to end of today's lecture. Do keep in mind what we discussed today. Time for you to self-study. If you want to learn more and enhance your knowledge, you may log on to our website, that is www.cec.nic.in. Till then, keep studying. Goodbye.